So we simply compare the motif regions of the ribosomal RNA to different organisms, and comparing them, we're able to generate what's called a cladistics-gram, which will enable us to determine essentially how closely related these organisms are. So essentially what you have is the more similar the sequence of the nucleotide base pairs that exists between the ribosomal RNAs, the more related the organisms are. And like I've said before, the reason we use ribosomal RNA is because it's very conserved. It's a very conserved type of genetic material. So there's not as many mutations as you would see. And so when you do see a mutation, that's indicative of evolution taking place. So on this cladogram, what you essentially see is that you have some organisms that are very closely related, more so than others. So like S. pyogenes and S. agalactia, and S. pneumoniae and S. mitis. These all are very closely related to each other. S. pyogenes and S. galactiae are more closely related than S. pneumoniae and S. mitis, again based on the 16S ribosomal RNA comparisons. But when you compare these organisms to E. coli and Shigella flexneri, which are down here in the cladogram, they have evolved from each other in a lot more grander scale. So the parents I guess the evolution of these organisms, what you see in this cladogram is that all of these organisms on this clade are all closely related. But the organisms down here are not as closely related, because you can see that there's a split. Anytime you see intersection here, 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 and here, what that means is that you have a common ancestor that exists between this group here and then these groups down here. The common ancestor has evolved or diverged at some point on the cladogram, and it gave rise to two other lineages. And then as you go further down the cladogram, such as the S. pyogenes and S. agalactia, you see another split. So here you have a parent, a common ancestor between these two, but somewhere the two evolved from each other and you end up with two clades that exist. So that's how you read a cladogram, which are very useful in helping us to understand the relationships between organisms in terms of evolution. So this is going to bring us to our conclusion of this chapter. It's a lot of information, I think, regarding the different structures. I think what would help many of you to further understand, especially the differences between prokaryotic cells and eukaryotic cells, is to spend some time making diagrams for yourself. You can simply refer to this PowerPoint or the images in your book and just make some diagrams or make yourself a T-chart to understand the differences between them. And then also make sure you understand the functions of the different structures that we highlighted, as well as how relationships among organisms in terms of their evolutionary lineages are analyzed. Okay? All right, well, thank you guys for listening, and I will post the next lecture next week. Have a good week.